Thank you, Colleen, for joining us today. We have this translational bipolar research rounds, and it's so nice to be able to get these um, expert perspectives on topics that are so germane to bipolar disorder. So Dr. McClung is a professor of psychiatry and clinical and translational science at the University of Pittsburgh School of Medicine, and she has a research program that focuses on the role of circadian rhythms in the development and treatment of psychiatric disorders and addictions. Um, it was during her graduate training with Eric Nestler, or postdoctoral training, I should say, that she transitioned her work to mouse models and conducted some of the first large-scale gene expression experiments using tissues from individual brain regions of mice, and became a leader in transcriptomics and the molecular biology of both addiction and mood disorders. We're going to hear about the multimodal research that she conducts with sophisticated analytic methods. But recently, her work has focused on treatment development as well, in particular treatments that engage circadian rhythms. Her work has been prolific and also recognized by a number of major national and international awards, including from the Brain and Behavior Research Foundation, Daniel Friedman Award for Outstanding Basic Research, the Colvin Prize for Outstanding Mood Disorders Research, McKnight Foundation Brain Disorders Award, etc. She's also a research leader, having been appointed to the Brain and Behavior Research Foundation Scientific Council, as well as the steering committee for BD Squared and an elected member of council at ACNP. Colleen, thank you for joining us. Over to you.
Yeah, thanks so much for having me. And yeah, you get to hear more about circadian rhythms, I suppose, today than you might have bargained for. But I'm excited to share some of our data. And because this session is really focused on genetics of bipolar disorder, I decided to really to keep it very gene focused and very genetics focused, and trying to understand how circadian genes are involved in regulating mood and reward and things like that that are associated with bipolar disorder. So first, a little bit of background about why we know circadian rhythms are involved in psychiatric disorders. So there have been a multitude of studies now that have shown that shifted or blunted circadian rhythms, as well as a late chronotype, so being a night owl versus a morning lark, strongly associate with multiple psychiatric disorders, including bipolar disorder. We also know that disruptions to the circadian clock via shift work, jet lag, seasonal changes can trigger or exacerbate mood and psychotic episodes. What I'm going to tell you more about this in a little bit, but there are polymorphisms in circadian genes that are associated with psychiatric disorders. And we also know that targeting circadian rhythms has therapeutic effects for some individuals. So, for example, things like bright light therapy, dark therapy for mania, interpersonal and social rhythm therapy are all treatments that directly target the circadian system. So this is just kind of a little illustration of this where you can have both environmental disruptions to the circadian system via a lot of different factors. And if you have some sort of genetic predisposition to a psychiatric disorder like bipolar disorder, these are just going to make it worse. It's going to really set people into episodes much more quickly, and make it much more difficult to balance that mood state across a long period of time. But there's also genetic mutations directly in circadian genes that can increase susceptibility to bipolar disorder and other psychiatric disorders. So at the molecular level, the circadian clock is really a transcriptional translational feedback loop. So it's regulated by the protein clock. Can you guys see my pointer here? Maybe, yeah, okay. It's regulated by the protein clock, or there's a homolog NPAS2 that is very similar to clock. It can bind to the protein BMAL1, and these act as transcriptional activators of the period and the cryptochrome genes. Then once these proteins are translated, they can form complexes that go back into the nucleus and inhibit the activity of BMAL1 and clock. And then there's also secondary loops where BMAL1 and clock are regulating the expression of ROR alpha and reverb alpha, which again can feed back and regulate expression of BMAL1. So these citations here are citations from different candidate gene genetic studies that have found mutations or polymorphisms that significantly associate with bipolar disorder. So you can see that some of these are in the ROR and reverbs or in BMAL1 or in clock. And so these smaller sort of candidate gene studies that have been done over the years have implicated the circadian genes in a risk for bipolar disorder. In addition, there have been some GWAS studies, so some larger genome-wide association studies, which have also found polymorphisms in circadian genes that associate with different psychiatric disorders. This was a nice study that was done by Mike McCarthy et al., which looked at several different GWAS studies and looked at both the core clock and genes that are involved in modulating the clock and also genes that are directly controlled by the clock and are really like known clock output genes. And so in the core clock, they found several different genes that had significant p-values in different GWAS studies that, for example, ARNT-L, which is the same as BMAL1, that associate with bipolar disorder, but also ADHD, schizophrenia, etc. And then when they look at the proportion of mutations that can be found with the core clock in both bipolar spectrum, but also lithium responsiveness, they find a greater amount than a random control. So this was also a really nice illustration of how these circadian genes can have polymorphisms that associate with psychiatric disorders. Another gene that's been highly implicated in bipolar disorder and schizophrenia is this gene CACNA1C. So it comes out in almost every single genome-wide association study of these two diseases. It's a calcium channel subunit, an L-type calcium channel subunit. And these Manhattan plots here kind of illustrate where you can really see this CACNA1C. And it turns out that CACNA1C is also involved in modulating the circadian clock. So CACNA1C is involved in photic entrainment. So the circadian clock, the master pacemaker, is located in the base of the hypothalamus in an area called the suprachiasmatic nucleus, or the SCN. And the SCN is basically innervated by retinal ganglion cells from the eye that are responsive to light. So basically light comes into the eye. There's projections from these retinal ganglion cells to the SCN. And then that can set the timing of the rest of the brain, the rest of the body, and help to entrain to the environment. And when light comes in, it activates both glutamate receptors, but also these CACNA1C channels in the SCN. And this can lead to phosphorylation of CREB. And CREB is directly involved in regulating the period and the cryptochrome genes along with clock and BMAL1 or ARNT-L. So this is just one illustration of how CACNA1C is involved in photic entrainment. And this is a paper that came out recently, which found that basically if you have this protein called GEM, where if you have activation of this L-type calcium channel, these CACNA1C related calcium channels, and you get this signal from light, that GEM is this protein that kind of turns it off. And so normally, as you guys know, if you've ever traveled anywhere overseas or, across different time zones, it takes a while to adapt to a new light-dark cycle. And that's because of GEM, because GEM is kind of inhibiting that L-type calcium channel. And if you take that away and you have too much of that calcium channel, then you just instantly entrain to the new light-dark cycle. So So this shows again how important those calcium channels are in the entrainment to light. Also, CACNA1C looks to be involved in regulating circadian rhythm amplitude and the response to lithium, even in fibroblasts. So if you take just skin cells and culture them, and you knock down the expression of CACNA1C in those cells, you can look at molecular rhythms through these reporter genes. These are luciferase reporter genes that have a circadian gene promoter. Then normally what you see is that lithium will increase the amplitude of expression, so kind of the strength of the rhythm. If you have a knockdown of CACNA1C, you actually get an increase in the rhythm amplitude just at baseline. But then if you have lithium, you get a decrease in that rhythm amplitude. So you can see it gets smaller. And you see some more subtle effects maybe from some of these other CACNA genes too. But it's really interesting that one of these genes that's been, very highly replicated to be associated with bipolar disorder and schizophrenia, this CACNA1C is also really involved in regulating the circadian clock in multiple ways. There have also been a number of studies of treatments for psychiatric disorders that have shown that they directly influence members of the circadian clock. So antidepressants, lithium, valproic acid, etc. All of these different drugs can influence members of the circadian clock either directly or indirectly, and can influence things like the rhythm amplitude or the period, so the length between the peaks of that circadian rhythm. And so it really does seem like the circadian clock is very important in both potentially the risk for bipolar disorder and also in treatment for bipolar disorder. If you can target these circadian genes, then you might be able to really enhance treatment. And that's something our lab is interested in doing. But mostly what I'm going to talk about is work that we've done to try to figure out how the circadian genes are involved in mood regulation. And we've done this through the use of a number of different mouse models. And so the mouse that we've used the most is this clock delta 19 mouse. So this is a mouse that has a point mutation in the clock protein. It was originally created by Joe Takahashi and Fred Turek and Martha Vitaturna. The point mutation was actually induced by a chemical mutagenesis, an ENU mutagenesis. And then they screened for mice that had circadian rhythm phenotypes. And this was actually mouse number 25 of a screen of thousands and thousands of mice. So they were very lucky to pick up this mouse. And they found that it had a long circadian free running period. But we, over the years, tested these mice in a variety of behavioral assays and found that basically in every way that we can test them, they have a very similar phenotype to bipolar mania. So they're very hyperactive, they sleep less than wild type mice, they have less sort of depression-like behavior or behavioral despair in assays like the forced swim test and the learned helplessness test. They have increased impulsivity, novelty seeking, risk taking in a number of behavioral models. And they're more sensitive to the rewarding effects of everything. So cocaine, sugar, brain stimulation, alcohol, you name it, like these mice really love it. And if you give them both either lithium or valproic acid, we can reverse those phenotypes and make them look more like normal mice. Excuse me. So these mice have become a really useful tool in our lab, both to study, how circadian genes are involved in regulating all of these phenotypes and also looking for treatments. So because they had this kind of hyperhedonic phenotype and this increased exploratory drive, and we really wanted to focus on the dopamine system. And so we decided to look at the ventral tegmental area, the VTA. And this is houses a number of the dopamine cells that are important in reward and motivation and these kinds of processes. So when we did electrophysiology, we did patch clamp electrophysiology and slices of the brain. We found that the clock mutant mice have an increase in dopamine cell firing in the VTA. And when we gave them lithium, this actually reversed their increased firing rate. So they seem to be hyperdopaminergic, and then lithium restores this to wild type levels. We then wanted to know, is it really clock in the dopamine cells that's causing this phenotype? And so we took a strategy where we knocked down the expression of clock, specifically in the VTA, and we did the same kind of recordings. And we found that, yes, indeed, if we knock down clock in the VTA, we see an increase in dopamine cell firing. So this suggests that it's not really clock in other regions of the brain or in the SCN that's causing this phenotype. It's more localized to clock in the VTA. So clock is a transcription factor. So we did some gene expression studies, trying to figure out what genes were changed in response to this mutation in clock. And one of them that we came up with was tyrosine hydroxylase. So tyrosine hydroxylase is the rate limiting enzyme in dopamine synthesis. And it turns out that the tyrosine hydroxylase promoter has an E-box, which is the binding site for clock. It also has a crease site, which is a binding site for CREB. And when we have this clock delta 19 mutation, we have an increase in tyrosine hydroxylase compared to wild type mice. And you can see normally there's this beautiful rhythm in tyrosine hydroxylase, but there's a sort of a shift and an increase in the clock delta 19 mice. And it turns out that clock directly binds to the promoter, kind of an antiphase with CREB. So the two of them will compete at that promoter to regulate this rhythm and activity. I'm not going to go into much of this today, but if you're interested, we did a whole study really looking more at the nitty-gritty of how clock is regulated at the TH promoter. It turns out that it involves this kind of sensing of the redox state of the cell, so kind of the metabolic state of the cell. And that's through the binding of this protein called sirtuin-1. And so this is really cool because it shows that, depending on like the metabolic state of the cell at the time, there's this signal that leads to an interaction with clock and regulates basically the amount of dopamine that's being produced through this clock mechanism. So if you're interested in this, you can check out this paper. So we also wanted to look at dopamine over different times of day. And so to do this, we did in vivo electrophysiology and recorded from dopamine neurons across the 24-hour cycle. And what we found that kind of matched that pattern of TH where we saw this increase during the daytime as opposed to the nighttime. We saw the same thing with dopamine cell firing and dopamine cell bursting, where we saw the biggest increase in the clock mutant mice in the daytime. So ZT0 is lights on. So that's about 7 a.m. in our mice. And they showed the biggest increase then. And mice are nocturnal, of course. So this is the time of day when they should be sleeping. But this is where they're having this big surge of dopamine at this time of day versus the active phase at night when the tonic firing is not that different. The burst firing actually is still different at this time of day. But this also kind of matches their behavior where they see the biggest sort of precipitation of that manic-like behavior during the day when they're supposed to be sleeping as opposed to during the night. So to test more specifically the timing of this dopamine surge, we turned to using optogenetics. So in this case, we're using a step function opsin, which is a little bit different than a traditional channel rhodopsin. Here, if you hit this opsin with a light pulse from the laser, it leads to a kind of a lasting increase. So of about 30 minutes as opposed to milliseconds. So it allows you to do more chronic experiments. So what we did is we basically took Th-Cre mice. So these are mice that will express the channel rhodopsin only in dopamine cells. We gave 7 days of stimulation, either during the daytime or during the nighttime. And then we tested them in a number of behavioral assays that are kind of looking at exploratory drive and anxiety-like behavior in these animals. When we, after this paradigm, when we looked at daytime stimulation or nighttime stimulation, we saw kind of an increase in just general locomotor activity in both of them, which you might expect when you have increases in dopamine like this. But when we looked at the elevated plus maze, which is a measure of this kind of risk-taking behavior or exploratory behavior, where the mice will either go into the open arm or the closed arm of this elevated maze, we found that the daytime stimulation led to more open arm entries in um, in these mice, both the time and the number. And the nighttime stimulation really didn't do anything. And we replicated this in the dark light test and the open field, which are very similar kinds of tests that measure like the mouse's desire to go into kind of a scary environment versus staying in a safe environment. And we found that daytime stimulation led to the mice really wanting to go more into the light, which is a scary environment for a mouse, and more into the center of these assays. And the nighttime stimulation had no effect. So what this really was showing us is that the problem is not too much dopamine necessarily. The problem is too much dopamine at the wrong time of day. So if you have a dopamine surge at a time of day in which the circuit is maybe not expecting it because the mouse is supposed to be asleep, um, then that's over time, that's what's causing these sort of pathological behaviors. If you just give excess dopamine at a time when the mice are normally running around, normally producing lots of dopamine, it really doesn't have any effect. And so this gives us a lot of clues as to how, things like circadian rhythm disruption, where people lose sleep and they are up, in irregular cycles. This could precipitate a manic episode because you're getting these surges of dopamine at the wrong time of day. To test more specifically about tyrosine hydroxylase, we gave a pharmacological inhibitor of tyrosine hydroxylase, this drug called AMPT. And again, these are just a number of different behavioral assays. And what you can see is if we gave this drug during the daytime, so this is going to reduce that abnormally high level of tyrosine hydroxylase, presumably reducing dopamine synthesis, then you're able to normalize behavior in like the open field, the elevated plus maze, etc. But when we give this drug during the night, we again have no effect. So again, really emphasizing this idea that it's not necessarily about the amount of dopamine or the amount of synthesis, it's about the timing of having it at an inappropriate time of day. So the conclusions of part one here are that circadian gene mutations are found in psychiatric disorders, and we know as well as genes that influence the function of circadian rhythm. So both the core clock genes and genes that can affect it, like CACNA1C. Circadian rhythms are targets of drug use to treat psychiatric disorders. Mice with a mutation in the clock gene have a behavioral profile very similar to mania, which is rescued by lithium and VPA. Clock seems to regulate tyrosine hydroxylase and the activity of dopamine neurons. And this activity is dependent on the cellular metabolic state. And dopamine surges at the wrong time of day seem to be really responsible for their precipitation of manic-like behavior. So moving on in the brain, so the VTA is doing a lot, but one of the things it's doing is projecting to the nucleus accumbens. So the nucleus accumbens is another real like center for emotional regulation, basically, and it receives these direct projections from the VTA. So we also wanted to look at what clock genes were doing in the nucleus accumbens and how the nucleus accumbens in general is regulated over the 24-hour cycle. So one of the experiments we had done a while ago was just do some more patch clamp recordings of medium spiny neurons now. So these are GABAergic neurons that are in the nucleus accumbens. And we found that there was this really dramatic difference between activity in the light and the dark, where we saw much more activity of these neurons during the dark versus the light. But interestingly, we didn't see any difference with the clock delta 19 mice, which suggested that maybe it was a different circadian gene that might be involved in these responses. And so we turned to this protein NPAS2. So NPAS2, as I mentioned, is very similar in function and in sequence to clock. It also binds to BMAL1, but it has this more limited pattern of expression. So clock is really expressed everywhere in the brain. Almost every cell has clock. But NPAS2 has this really strong expression in the nucleus accumbens and also in the dorsal striatum. It also has expression in cortical regions as well. But you can see we were really intrigued by this like huge expression in the nucleus accumbens. And excuse me, the nucleus accumbens is it's about 95% medium spiny neurons, so those GABAergic neurons, but they kind of come in two flavors. So one of them has expressed the dopamine D1 receptor, and these neurons form the direct pathway, which project back to the VTA and in the dorsal striatum into the substantia nigra. Or they express the dopamine D2 receptor, and these form the indirect pathway, which go through the globus pallidus and then back into the VTA. So they're really these neurons are very distinct in terms of their a lot of their molecular signatures and things that they do. The D1 receptor is will stimulate the second messenger pathways. The D2 receptor inhibits them. So we really want to look at these two different subtypes of neurons in the nucleus accumbens. And when we just looked at clock and NPAS2 expression in these neurons, we found that NPAS2 is very highly enriched in D1 neurons as opposed to D2 neurons, whereas clock is kind of much more even between the two. So then we took the approach again of knocking down the gene. So we knocked down NPAS2. And we found that when we knock down NPAS2, that this, and we record, again, do patch clamp recordings onto the medium spiny neurons, we find that this leads to an increase in kind of glutamatergic activity. So kind of an increased just overall activity pattern of the D1 neurons, but not the D2 neurons. You can see that most readily here in this AMPA to NMDA ratio, which kind of means more activity. And so it seems like NPAS2 is really not only expressed mostly in D1 neurons, but also impacting the activity of D1 neurons much more strongly. We also wanted to look at some behavior. Here we were doing a cocaine preference test where we were looking to see if mice prefer to be in an area where they've had cocaine before versus not. So this is a measure of reward. And we knocked down NPAS2 either in D1 cells or in D2 cells. And we found that when we knocked down NPAS2 in the D1 cells, we actually saw a decrease in cocaine preference, where if we knock down in the D2 cells, we see no effect. And so this is interesting because this is, very different than the clock mutant mice that have this increase in cocaine preference and increase in um, reward value. Here, when we're knocking down MPAS2, we're actually seeing a decrease in cocaine preference. So then we wanted to kind of take a step back and look at the overall kind of molecular signatures of these neurons. So here we took an approach where we did a ribotag assay. So these are mice that have a tag on their ribosomes. And you can mate these mice with different Cree drivers, which either will express that tag in D1 cells or D2 cells. And we also did it in astrocytes, so in glial cells. And then you can pull down specifically the ribosomes along with the RNA that's bound to those ribosomes. So it's the RNA that's being actively translated with those ribosomes. And then we can kill mice at different times of day and do RNA sequencing and look at basically what kind of transcriptional profiles we get. So when we just look at this in general of all the genes that kind of have rhythms in 24-hour rhythms in these cells, which there's quite a few of them, we see that in both D1 and D2 cells, there's these kind of two peaks of expression where genes are either peaking during the day or they're peaking during the night. Interestingly, astrocytes, we found that more of the genes are peaking at night than during the day. We also looked at sex differences to see if there was potentially any kind of sex difference in the genes that had circadian rhythms in these cells. There may or may not be. We weren't really sure. But basically, when we look at the genes that are rhythmic in males and females, and we use this RRHO, or this rank-rank hypergeometric overlap, basically what this does is it just compares two different like lists of genes that to see how similar they are, and it does it in kind of a threshold-free way. And so the more red means the more similar. So you can see here that on the right here, when you look at D2 cells and we compare males and females, you can see it's almost all red. So that means that the same genes have circadian rhythms in males and females. There's really not much difference. But in the D1 cells, interestingly, there's not as much overlap. So it means that there's really a big sex difference in D1 cells specifically. Where the genes that are rhythmic in females are not necessarily the same as the genes that are rhythmic in males. And when we look a little bit more at the timing of this with these radar plots, what we find is that, again, there was this sort of general trend towards genes either having peaks during the day or peaks during the night. But these genes that are uniquely rhythmic in D1 cells in males have like a totally different timing of their peaks, where they're either peaking kind of at lights on or lights off. And so we really don't know what this means, but it's just kind of interesting that these cells have these very specific patterns of rhythmic gene expression, and that in D1 cells there tends to be this really interesting sex difference. And this is just kind of an overview of some of the major processes and what time of day they're being regulated in males and females in these cells. So things like synaptogenesis, protein folding, immune responses are, somewhat different, especially in these D1 cells. We also recently have been doing recordings, again, patch clamp recordings, specifically in D1 or D2 cells at different times of day. And what we're finding is that D1 cells and D2 cells tend to have this very opposite pattern of activity where they're basically in anti-phase with each other. So when D1 cells are active, D2 cells are inactive across a 24-hour period. And this is, again, really important in trying to figure out how these cells are modulating reward-related responses and mood-related responses. Because you can imagine if this delicate pattern gets out of whack, this could really lead to changes in behavior, since if you activate D1 cells, you get kind of an increase in reward, whereas if you deactivate or if you activate D2 cells, you get a decrease in reward. And then we also wanted to look at the glial cells. So we looked at these astrocytes in the nucleus accumbens. They're very beautiful cells. They're these star-shaped cells. And they're really important in supporting basically the metabolic state of neurons. They're very important in providing energy for neurons, cleaning up byproducts of neurotransmission. So astrocytes have a lot of really important functions. So one of the things we found when we did this kind of transcriptional profiling of astrocytes is that they are incredibly rhythmic. So almost 50% of their genome had a circadian rhythm. They were very, very rhythmic. And you can see when you look at the expression of these core clock genes, they're exceptionally strong. So the circadian rhythms in astrocytes are probably really, really important. Interestingly, we see the same thing when we do human brain. We do studies in human brain. Astrocytes also have very, very strong rhythms. Microglia and oligodendrocytes tend to not have as strong rhythms. So there's something very specific about astrocytes and how they function, where they really need strong rhythms. So again, getting back to like, what is the molecular clock doing in these cells? So here we took an approach where we knocked out BMAL1. And so the reason we went to knocking out BMAL1 as opposed to clock or NPAS2, this is kind of a sledgehammer because BMAL1 binds to both proteins. And so if you knock out BMAL1, you're going to get rid of either function of NPAS2 or clock. You're just going to disrupt the molecular clock in general. And so we knocked out BMAL1 specifically in astrocytes in the nucleus accumbens. And when we did a battery of behavioral tests in these mice, what we found is that this produced this increase in exploratory drive. So this is kind of similar to what we see with the clock mutant mice, where in the open field, in the like dart test, in the elevated plus maze, novelty suppressed feeding test, if you test them during the day, you get this kind of increase in like anti-anxiety, exploratory drive, risk-taking behavior, whatever you want to characterize it as. Interestingly, again, you test them during the night, we saw no effect. So the timing of all of this is super, super important. We also wanted to look at motivated behavior. So we did operant food self-administration. And when we tested them just on an FR1 schedule, so where they had to press the lever one time to get a food pellet, we saw a sex difference, but we didn't really see much effect of the BMAL1 knockout. But when we challenged them a little bit more, make them press, 10 times to get one food pellet, then we started to see that the BMAL1 knockout in astrocytes led to more motivation for that food reward. Again, only if you test during the day, not at night. And also, we recorded from nearby neurons. And so knocking out BMAL1 in the astrocytes actually impacted the activity of nearby neurons. So it decreased this glutamatergic drive onto these neurons. So this is kind of the opposite of what we saw with the NPAS2 knockout, which makes sense in terms of the behavior that we're seeing too. And this was interestingly very similar to what we saw when we recorded in the nucleus accumbens of the clock mutant mice, where we saw this decrease in AMPA NMDA ratio, which suggests that maybe what clock is doing in the nucleus accumbens is most important in astrocytes, whereas what NPAS2 is doing is most important in D1 cells at least. And then D2 could be either one, could be both. We're not really sure. Okay, so the conclusions of part 2, we find that NPAS2 is important in the nucleus accumbens, particularly in these D1 cells in regulating reward and neuronal activity. D1 and D2 cells have different molecular signatures, and especially the D1 cells seem to have a sex-specific signature. D1 and D2 neuron activity is opposite in phase. And astrocytes are incredibly rhythmic. They're incredibly important. If you knock out the molecular clock in astrocytes, then you get changes in behavior and changes in neuronal activity. And so, by kind of taking both together, like trying to understand what these clock genes are doing in this VTA nucleus accumbens cycle and in these specific cell types, hopefully this will get us a little bit closer to trying to figure out if someone has a mutation in these genes or, has disruptions to the circadian system via an environmental disruption. How does that contribute to psychiatric diseases and in particular bipolar disorder? So I will stop there and thank, this is the current lab, and thank also my funding and some of the past members of the lab that I listed here that have done a lot of this work, and open it up for questions. So thank you.
Thanks so much, Colleen. So much data, so much to think about. We'll just let folks either unmute themselves or type their question in the chat.
Yes. I see one hand up, so yeah.
And then go ahead.
No.
Oh, sorry. I think it was Dr. Milson.
Want me to talk?
Yes.
Go ahead, Benoit.
Okay, so first, welcome and thank you for doing this seminar. It's fascinating. It is great. I do believe we need to do more animal study to bridge between the cell and the behavior, if you want. The behavior is an intermediate level between the cell, the animal, and the human. So I really appreciate your work. However, okay, and I don't want to be a Debbie Downer and I'll be unpleasant, but your lecture reminds me of 10 years ago, there was a brilliant guy who was doing epigenetics, or 15 years ago. We all thought the epigenetic will be the key to something. And that person got a chair in Toronto. And then we did a presentation at the end of his first term. And the big discovery is that there's an epigenetic clock. So the epigenetic factors are different at 8 in the morning and 4 in the afternoon.
Yeah.
And I realized, oh boy, okay, we are not going to get an epigenetic answer. Since we didn't get the genetic didn't solve, or now we have the gene for schizophrenia or bipolar or whatever, or for the lithium responsive subgroup of patient on bipolar. Then I thought maybe epigenetic will solve the puzzle. And then the guy said, good news, we have a big discovery, bad news. It's way more complex than we thought. There's an epigenetic clock, and which means every study that has been done on epigenetic of psychiatric disorder has to be redone. Because if you drew the blood at 8 in the morning or at 2 in the afternoon, and you analyze the data themselves, it means nothing. Okay. And I say, oh boy, okay. So hearing you are telling us, the simple model that I was taught when I was a resident of monoamine and this and that, you say, it's way more complicated. It's not just, I heard your sentence, it's not just dopamine. It depends at which time of the day the dopamine works. So my question is, it's great for your lab. I'm happy, we can keep brilliant scientists like you and your PhD busy for the end of time. Do you really believe that, and I'm too old, during my professional lifetime, no, but for the people in this virtual room who are in their 30s and late 20s, do you believe that we will be able to do the bottom-up bridge and go back from the kind of research you do with cell to clinician like me to help us manage our patients?
Yeah, I know. It is. I feel like you're not alone. And whenever I talk about these kinds of circadian things, people are like, oh my gosh, like the time of day is so important and we haven't thought of that at all. And yeah, it always like freaks people out. Like we have to do our behavior multiple times a day and da da da. But actually, yes, because I feel like there's already clinical evidence that if you just stabilize the clock in any way, that it's going to be helpful, right? So, I mean, we're trying to figure out the nitty-gritty of like how these things work in the brain, but it's possible that at the end of the day, all you really have to do is keep that clock stable, and that's going to be, really helpful and therapeutic for a lot of people. And for some people, we might need to think about shifting one way or shifting another way, or, something like that. But from studies of like interpersonal social rhythm therapy, for example, which is just a cognitive behavioral therapy where people are put on a very strict sleep, wake, and social schedule, that's really helpful for people with bipolar disorder. And we're trying to come up with medications that target the circadian system that are going to, amplify those rhythms the same way that lithium and valproic acid do. And, but in a more targeted way. So yeah, so I think there is, some hope that even though this is really complicated, that, even just knowing that if you can stabilize the clock, it's going to help. I think that's still good. Yeah.
Thank you. This is reassuring. And I'm from Pittsburgh,
Oh,
like
good.
like
Okay.
like Ben Goldstein, or it's also spent some formative year there. So we
Yes.
all know about Elaine Friend and her ISPRT.
Yeah,
So
yes, yes, yes.
that's where it comes from.
I know, yes. Yeah, yeah. Ellen Frank and David Kupfer, yeah, they really are the ones who pioneered a lot of this field. Yeah.
Man, did you stop want to ask a question?
Should I stop sharing, or should I so I
Sure.
can see people? Okay. Yeah. Okay.
So there's another question in the chat from Jim. Did you want to? I can read it out. Let's see here. Actually, I'll go in order here. We have Andrea Vasilev who's asking, how does this research map onto genetic risk of an individual with bipolar passing on genes loaded for bipolar? For example, are there circadian phenotypic attributes or their seasonal patterns, susceptibility to episodes during circadian changes, etc., that indicate an individual has a heavier genetic load or is more likely to pass on their bipolar disorder genes?
Yeah, that's interesting. There haven't been a ton of studies that have really looked deeply into these kind of circadian like endophenotypes. I know that's something that people are really interested in doing. So I'm part of the BD squared network. From the Milken Foundation, and we're doing like a huge longitudinal study across multiple sites. And one of the things they're really interested in looking at are these sort of circadian factors and how they might relate to genetic factors, and yeah, whether like seasonal patterns that are very common in bipolar disorder, right? It's very common that people will have mania in the spring and depression in the fall and winter, whether that's something that runs in families. And it seems like it does to some extent, but honestly, like there just haven't been enough studies to really definitively say like, aha, like if you have this circadian pattern that goes with these genes and it's passed on through the families, but it's something that's worth studying for sure.
Colleen, I'll take a shot at a question. So, like Benoit, I think I'm interested in the translational potential. And one of the things that interested me was just the intersection between sleep, reward. Um, and I'm wondering, with the increasing emphasis of on energy and mood disorders in particular, and then you're talking about consummatory behavior, physical activity, sleep, like to what extent are there individual differences in the extent to which those things are constrained together versus independent? Like, can you have normal eating, normal energy, and then have a sleep circadian dysfunction, or how does that work in humans?
Yeah, it seems like they sort of go together, right? I mean, like it's a lot of times when people have like sort of atypical depression, right? They have like overeating and oversleeping, or um, people when they have seasonal depression will have this kind of again like overeating and especially carbohydrate eating and and lots of sleeping. So it seems like those kind of patterns tend to go together unless, yeah, and then the same with like insomnia and not eating as much. So yeah, they tend to sort of go together. There's a very intricate relationship though between the circadian clock and metabolic function, both sort of in cells and also just in the body. So, there's a lot of interest in how disruption of circadian rhythms really impacts your insulin signaling and metabolic syndrome, and your clock in the liver is very important. So yeah, there's a lot of interactions there between the circadian clock and metabolism.
Just a follow-up question to that, and then there's another one in the comments, which is withholding sleep or interrupting sleep is thought to be like a pretty replicable strategy for highly treatment-resistant depression, often done in hospital settings. Has
Yep.
anyone studied like the biology of that in a similarly elegant fashion to what you've done in the mice, or can you do that in mice?
Yeah. So people have done some studies of this. It does look like, yeah, sleep, like acute sleep restriction kind of leads to this hyperdopaminergic phenotype. And it also actually has a lot of similar genetic signatures to ketamine treatment. So if you give ketamine, which is a rapid acting antidepressant, and you give acute sleep restriction and do RNA sequencing in mice, you see very similar kind of molecular signatures. And they're a lot of the circadian genes are actually like changed by these both of those sorts of manipulations. So I wouldn't, there hasn't been quite as much sort of real analysis of like the cell type specificity or any of that type of stuff, but it definitely, both of them tend to lead to this kind of like increase in BDNF, increase in plasticity, increase in dopaminergic signaling, but it doesn't last, right? So if you once people have they go back to sleep, especially if you go into REM sleep after having acute sleep restriction, the depression comes back. So it's definitely a very short term kind of treatment.
So the next question kind of builds along this as well. This is from a lived experience perspective, asking the question of whether medications that have a beneficial effect on sleep in terms of causing or at least constraining it. So with clozapine, is the sleep control that comes from that medication or its sedative effect, does that contribute to stability in circadian?
Yeah, there hasn't been a lot of evidence that I think just giving people sedatives really can like help to keep the circadian clock, regular. You know, um, it's more about, yeah, the time, having the regular timing of sleep than the, just knocking someone into sleep or having a long period of sleep. It's so there hasn't been a ton of evidence suggesting necessarily that like if you gave someone Ambien or whatever, that's going to be a good treatment for bipolar disorder.
But if you do it at the same time every night, would it?
I mean, maybe that would help some, but yeah, like, people have studied melatonin, for example, also. And yeah, melatonin has some effects, but it's not the greatest um at really influencing bipolar disorder. So yeah, I know there haven't been a lot of studies that have suggested that that's so as good as some of the other treatments that really target the circadian clock more specifically.
So, and then Anthony Levitt's asking about whether it's dichotomous night versus day circadian rhythms, or are there within daytime, or I guess within nighttime variability in circadian rhythms?
In bipolar disorder?
The question's in general, but however you want to take it.
Well, I could say that like when people have studied circadian rhythms in people with bipolar disorder, like through actigraphy and those sorts of studies, that their variability is the most common thing that you see. It's really this like intradaily variability. So having this kind of variable sleep wake time during the day, but then also between day variability where you see a lot of up and down and up and down of different sleep patterns. And so those are the most common phenotypes that are found in when you do actigraphy in people with bipolar disorder. They also tend to be night owls, so definitely like to get up late and go to sleep late. That's very common as well. Also common in depression, but yeah.
Jim, last question over to you.
Yeah, so I had patient who was very a teachable moment was provided. So he had schizoaffective manic disorder. His family asked me if I would allow him to fly to Europe, to Croatia to meet his grandfather who was dying.
Hmm.
And I thought it would be fine, and I approved it. He went there. He developed florid mania, was hospitalized, put in restraints, given chlorpromazine, and broke his arm in the constraints, and his arm is still crooked to this day. So
Wow.
the question then, should we be paying more attention to this natural experiment of people with bipolar tendencies? And being a geneticist, I would say if they have a high polygenic risk score for bipolar,
Hmm.
they should be very careful when they change time zones
Yes,
to that
absolutely.
extent. And that's my anecdote, but I haven't seen a systematic study of patients who do trans time zone travel. There should be a much higher rate of induced mania.
Yeah, there have been some studies. There's not a lot. You're right. There was one study that looked at people when they were traveling through London Heathrow Airport and the number of hospitalizations and whether people were manic or depressed at the time of hospitalization. Really interestingly, if people were traveling eastward, where they were shifting their circadian clock forward, they were more likely to become manic. If they
Yes.
were traveling westward, where they're shifting their clock backward, they were more likely to become depressed. And there's actually some studies out of a lab in Korea that also took people who were hospitalized and whether they came in manic or depressed, and they were able to see a phase advance in rhythms during mania and a phase delay in rhythms during depression that then went back to normal during euthymia. And so, yeah, there are some studies out there that have looked at that, and the social Zeitgeber theory of bipolar disorder that was put forward by David Kupfer and Ellen Frank. This is exactly kind of what they postulate, is that these changes to the light dark cycle through travel or shift work or whatever, that this is what will precipitate episodes, and that's why you really need to keep on a very strict sleep wake schedule. So yeah, it really is something that I think physicians should be cautious about, because I've talked to so many people who have told me, they were fine until they traveled, or until they stayed up all night working on a project, or until they, worked a night shift or whatever, and then it was like boom, that was the trigger for their episode.
So thank you so much, Colleen, answering
Mhm.
all of these clinical and translational questions in particular, and also presenting your really elegant research so clearly to us. This closes out our year and this session. So thank you, everybody. Please submit your feedback to us, and in particular, if you could email us with any ideas, suggestions for topics you'd like to be see covered next year. All of these presentations are going to be available through our website, through a YouTube channel. So if you missed anything, it'll be there. But also, I'm keen to know what people want to see next year. So thank you, and have a great summer.
All right, thank you.